afternoon, everyone. So we're going to be getting started. My name is Alvisha Damala, and I'm an assistant professor at the Feinstein Institutes for Medical Research.
Hello, everyone. My name is Di Yiru from Institute of Statistical Science from Academia Sinica of Taiwan.
And we're both extremely excited to introduce our keynote speaker for tonight. So tonight we have Dr. Nicola Palomero-Gallagher. So she graduated in 1990 from the Sciences faculty of the Universidad Autónoma of Madrid, Spain, where and then she was a PhD student at the CNO Vought Brain Research Institute of the Heinrich Heine University in Düsseldorf, Germany, between 1992 and 1999, where she got her PhD. Since 2000, she's been working at the Institute of Neuroscience and Medicine at the Research Center Jülich in Germany, where she is a senior researcher and leader of the research group Receptors.
And Dr. Nicola Palomero-Gallagher, she is a senior editor of Brain Structure and Function, and her research is centered on structure, including the cytal and the fibrate architecture, and the molecular, the receptor architecture, organization of human, non-human primates, and the rodent cerebral cortex, and with particular focus on the cingulate cortex. And her major goal is to understand the cytal and the receptor architectonic basis of cortical segregation and the interareal intersections, and with emphasis on their relationship with function. So without further delay.
Yep, so today she'll be presenting her work on neurotransmitter receptors that link structural and functional segregation patterns in the brain. Please welcome her.
Good evening, everybody. Thank you so much for the wonderful presentation. Thank you very much for the organizing committee for considering me as a keynote lecturer. Max said yesterday that OHBM is a conference where he grew up. It's also the conference where I grew up. This here is a photo that was taken during my very, very first OHBM meeting. That's me there at the bottom right of the slide. I didn't present a poster or anything at that thing, but we played a football match against the Phil Institute, and we won. And if you told me then that I'd be giving a keynote lecture here today, well, I would have told you that you don't know what you're talking about. So anyway, let's get back to science, because we've only got 45 minutes. When people think about connectivity in their brain, the first thing that comes to mind is normally fiber tracts. And many people tend to forget that at the end of these fiber tracts, which are basically just a collection of axons, that there is a synapse, and that neurons are actually not physically connected with each other. So when you reach this synapse, the cells need some sort of a mechanism by which the signal can be transferred over the extracellular space. And this mechanism is called chemical conductance, and it is carried out by the neurotransmitters, which are symbolized here by the little green dots in the figure, and by their receptors, which are the purple or pinkish cylinders in the slide. Now, receptors are simply proteins or protein complexes, okay, and there are different methods that you can use to visualize these structures of the brain. And at the two ends of the slide, you can see the two extremes. You've got to the left of the slide a PET image that allows in vivo visualization of receptor distribution patterns, so a really good positive point of the method. It has a relatively low spatial resolution, and there's a limit to the amount of receptors that you can analyze with it. And on the other side of the spatial scale, you've got immunohistochemistry, which really, if you use electron microscopy, you can go down, into the nanomole range. Now, the method that I'm going to talk about today during most of my talk is the method that's presented here in the middle of the slide, quantitative in vitro receptor autoradiography. And it's a very powerful method because it is fully quantifiable. It provides quite a good resolution, enabling visualization of structures at the microcircuit level, and you can analyze very many different kinds of receptors. It has the disadvantage that it's post-mortem and it's very expensive and very time-consuming, but it produces beautiful images, colorful images. And this is the kind of images that you obtain when you do receptor autoradiography, okay? Normally, receptor autoradiographs are just gray images, different shades of gray, but we assign them a color scale to make differences in receptor densities easier to see. And what you here see are coronal sections through a human hemisphere, which has been, and each one of the sections has been processed for a different receptor. And the coding is such that blue tones code for low receptor densities and red tones code for high receptor densities, okay? And this is the color coding that I'll be using throughout my talk when you see a color-coded receptor autoradiograph. Now, even in this overview slide at this really low resolution, you can see that receptors have a heterogeneous distribution throughout the brain. And we've got regional differences, we've got laminar differences. And this regional distribution helps us to visualize, to analyze different organization principles in the brain. The first one of these organization principles is that differences in the receptor distribution patterns highlight borders between cortical areas. And when two or more receptors highlight this border at a certain position, it's at a comparable macroanatomic position, although please always remember that each one of these images has been obtained from a different section, okay? So what you here see is a cutout from one of the images that I showed in the previous slide. And it's centered over the primary auditory cortex, A1, and it shows the muscarinic M2 receptor, and A1 is characterized by a super high density of these receptors, and there where that red color disappears, that is actually the cytoarchitectonic border, or it corresponds to the cytoarchitectonic border of A1, and it's highlighted by the arrows. And if you look at this autoradiograph here and compare the position of the arrows, you can see that changes in the alpha-1 receptor also highlight the border of the primary auditory cortex. Only for this receptor type, it's characterized by a lower density than the surrounding areas. And then we've got receptors such as the NMDA, which don't show the border of the primary auditory cortex, okay? So not all receptors show all possible borders, but when several ones show it, they show it at a comparable position. The second organizational principle is the fact that receptors segregate functional systems, okay? And this is very clearly visible in this slide here. It's centered around the central sulcus, and the central sulcus is like the home of the primary somatosensory cortex, S1, and the primary motor cortex, M1, on this slide. And the interesting thing is that the receptor architecture of these two brain regions, which have really different functions, it's almost anti-correlated. When you've got a receptor type with a very, very high density of a given receptor, the other area normally has a very, very low density. Now, the next organization principle that I want to talk about is that it's not enough that receptors show borders and show functional systems, but within a given functional system, receptors also highlight hierarchical processing levels. And this is very clear on the image on the right of the slide, which is again a coronal section. It's through the occipital lobe of a human hemisphere. Again, it's the M2 receptor, and you can see that the density of M2 receptors decreases systematically when we move up the hierarchy within the early visual areas, so unisensory or unimodal visual areas. And the same thing happens for the auditory, for the somatosensory system. And finally, the method of quantitative receptor autoradiography is super helpful when you want to perform comparative analysis, because you can actually quantify the degree of similarity between different species. And that means that you can then decide if species A, for example, the macaque monkey, or your animal model B, the rat, if they are for that particular receptor type that you're targeting, or that particular brain region that you're interested in, which one of those two models would be the better one for you to use if you want to then be able to translate your results to the human brain, okay? Now, my team and I have been working for the last 5 years or so a lot on comparative analysis, building on this last aspect here of receptor autoradiography. So during today's talk, I'm going to be switching between results that we obtained in the macaque monkey and in the human brain, but whenever I switch from one species to the other, I'll let you know, okay, so that you're not hopefully confused. I'm going to start with the macaque monkey brain, and I want to present some work of my postdocs, my team. This here is, or these are maps of the frontal lobe of the macaque monkey brain, which Lucia has been working on, and it's truly impressive work. And the word impressive, it's not me being the absolutely unbiased supervisor. It's actually me citing David Badre, who was the handling editor of the eLife article. Then there is the work by Mei Qi. Mei Qi has been concentrating on the parietal lobe. She's also been doing visual cortex, and what I think is really nice, because we need this in the field, or this is at least my belief, Mei Qi is now also slowly acquiring computational expertise, and it is something that I believe that the neuroscientific community will need, that these different fields that we communicate with each other. And we also had a collaboration with Carmen Cavada at the Autónoma University in Madrid, and they have been mapping the thalamus brain, and our combined efforts have resulted that in the parcellation of over 130 cortical areas in the macaque brain and over 80 subcortical structures in the thalamus, the amygdala, the basal ganglia. Not everything has been published yet. And, you know, I could spend hours and hours and hours showing you different color-coded images and sections and pointing out, you know, fascinating relationships. But I've only got 45 minutes, and I only have 34 left, actually. And I think, you know, I'm not allowed to talk for longer. So I'm just showing you this selection here of slides through different rostrocaudal levels of the macaque brain, and which were processed for the visualization of the alpha-1 receptor, so that you can get a bit of a feeling for the level of granularity and the complexity of the patterns. If you are interested in these maps, we make them, once they're published, we make them freely available via the MeBrain's multi-level macaque brain atlas, which you can access via the QR code on the bottom of the slide. We also make them available via Balsa. And if you go onto the eBrains platform and you want to, you can actually switch from the macaque monkey brain, which is the one that this QR code would take you to per default. You can switch without any problem to the human brain and, for example, see big brain or see the Jülich maps, for which we've just released a new update with new areas. Okay, so it's exciting developments. Now, one of the advantages of receptor autoradiography is that it is a fully quantifiable method, okay? So you can, once you've identified an area, you can measure the densities of many different receptors in that area, and we chose to visualize these densities in a polar coordinate plot, and we called this a receptor fingerprint because it is unique for every brain region, okay? And receptor fingerprints visualize the balance of the different analyzed receptors within the brain region in question. And receptor fingerprints can differ in either size. So if ones are bigger or smaller, it means you've got more or less receptors in the area in question. And they can also differ in shape, meaning that the balance between the different receptor types is different in the areas. And this is important because some of these receptors are excitatory, other ones are inhibitory, some of them are metabotropic, ionotropic, so some of them have a slow mechanism of action or a faster one or a longer lasting one. Some of them bind to modulatory neurotransmitters. So this gives the brain the plasticity to be able to subserve function, okay? And what we found was that if you are comparing areas low down in the hierarchy, so let's say the primary visual area with secondary, tertiary areas, we already find differences in the fingerprints, and these differences are actually larger than the differences that you find between two related association areas. And the association areas, their fingerprints become sort of like flatter, rounder, because they have to be able to communicate with very many different areas from different functional systems. Now, fingerprints, we don't just have these regional differences in the receptor densities, we also have laminar differences. And we performed an analysis of laminar receptor densities across over 40 brain areas, compared it to the site architecture. And what I've done in this slide here, it's just two examples, V1, V2, and I've transferred the lines delimiting layer 4 over onto the autoradiograph to make a point clear, and that is that we don't have a one-to-one correspondence between what we call cytoarchitectonic layers, so the ones that you can define based on the distribution of cell bodies, and receptor architectonic layers, okay? But still, it's because the problem is that although, so there is my mouse, although these red densities here, these high densities here, are located in cytoarchitectonic layer 3, that doesn't mean necessarily that they are only located on the dendrites of layer 3 pyramids, right? Because we know that layer 5 pyramids, layer 6 pyramids, they can also send their dendrites up here, right? So this is something that we have to keep in mind and find a way also of working out maybe how to segregate these laminar differences. Anyway, we can also extract fingerprints from individual layers. And here each layer that you can see labeled at the top of the plot is indicated on the color-coded by its own color. And we found for most areas a canonical sequence whereby the superficial layers showed higher densities than the deeper cortical layers. And the only notable exception for this is basically the Keynote receptor, okay? And not surprisingly, this canonical sequence in receptor densities actually correlates pretty well with the synaptic or the laminar synaptic density, because we know that layers 3 and 4 are the cortical layers with the highest density of synapses. Now, we wanted to see if these laminar receptor fingerprints, a statistical analysis, if they would tell us something about global organizational principles in the brain. So we performed a principal component analysis using these receptor fingerprints, which can basically be treated as feature vectors. And when you look here at this, we performed a multidimensional scaling analysis, not a PCA. When you look along the dimension 1, you can see that the dimension 1 separates the fingerprints that were obtained from the superficial and the deeper layers and layer 4, and the colored surfaces in here are just meant to highlight that fact, right? Because they encompass all the colors of the corresponding layer, with the notable exception of the primary motor cortex and layer 6, where we did extract densities from a very thin stripe of cortex at the interface between layers 3 and 5, but the existence of a layer 4 in these areas is being, or in the primate brain is still discussed controversially. And then you've got, at the bottom of the plot, you've got two areas in black, the entorhinal cortex and the hippocampus. That is because they don't have these supragranular, infragranular, and granular layers. So that's why each one of these areas is represented by a single dot. Now I'm going to get rid of most of these dots in a second, because I want to leave a couple of areas to make a point clear. But just before I do that, in case I didn't make my point clearly enough, that the second dimension separates areas which are older from the evolutionary point of view, so the entorhinal cortex and the hippocampus, from those which are newer, okay? Right, so now I get rid of a whole load of dots, and I'm just leaving in here the dots that correspond to our primary sensory areas, V1, S1, A1, and then I've got an area which I've labeled parietal. It's area 7, it's a parietal association area. And the take-home message here is that the dimension 1 doesn't just separate, the different layer types, but it also segregates areas that are mainly involved in sensory processes from areas that are involved in association processes, right? Just the same as dimension 2 segregates the allo from the neocortical areas. Right, so I'm going to stay with the monkey, but I'm going to move over to a couple of projects that were born at an OHBM meeting, the one held in Vancouver, where Sean approached me and wanted to know if I'd be interested in a collaboration. At the time, he was a postdoc at Xiaojing Wang's lab, so, doing computational neuroscience, large-scale cortical networks, way above my understanding, but he's good at explaining things, and he wanted to use the receptor densities to inform his models, which I think is a good idea. So I said, yes, let's go ahead and do it, right? Now, as a spin-off of this main first question, we did another thing, and that was to look at the receptor architecture and circuit details of large-scale cortical networks, so at all of these different areas that we'd been mapping. And for this, we integrated the receptor density from all the areas that we'd been mapping at the Institute. We integrated them with data concerning neuron density, and we obtained that from data published by the CAS group. Then we also used dendritic anatomy data, which has been published by Elston and collaborators, but here I'm only citing his one book chapter because it sort of like collates all of his other articles, and there's so many that fill the slide here. And we also integrated it with tract tracing data obtained from the, or not obtained, produced by the by Henry Kennedy's group, and which provides a weighted and directed interareal connectivity matrix. So it allows us also to make inferences concerning hierarchical levels. And now I'm showing the result of a principal component analysis. And what you see here is basically the macaque monkey brain mirrors the situation that I showed you a second ago for the human brain, that the first dimension separates sensory from association areas, and the second dimension separates allocortex or from neocortex, okay? Now, today's lecture, I said I was going to try and integrate receptors and function. So here you've got function. Human function or cognitive neuroscientists normally or often define 6 or 7 cognitive networks, and each one of them is color-coded here with a different color, so the visual in a sort of a purpley and dorsal attention in green, and then the areas that have been assigned to each one of these networks are coded in the corresponding color. Okay, so this is meant to make life a bit more intuitive and easier for you to see. Now, two of these networks are mainly associated with sensory processes. They are the two with the top bracket, and the other ones are associated with cognitive processes. And when we try and say, okay, do our areas separate in some way, we see that areas that belong to the sensory networks, they are located more to the left of the plot, which isn't surprising given that the receptor principal component 1 separates the sensory from the association areas, whereas all the other areas are located then on the right side of the plot. And then if we concentrate on areas that are located between the square with the dashed lines, and you look at the top right of that image, you'll see a whole load of areas that are coded in red, and they belong to the default network, which is sort of like a pity to call it default, as if it weren't doing anything when you're doing important functions. It actually does quite a lot of interesting or it's involved in quite a lot of interesting functions, and it's also particularly active when you're not concentrating on the outside world, but you're concentrating on your inside. So then what we have here is, because the green areas, which are at the bottom of this plot, and they belong to the dorsal attention one, what we've got here is also that we've got a separation, between the sensory and cognitive, and we've got on the second component, we've got a segregation of internal focus versus external focus, okay? Now, let's have a closer look at this sensory cognitive axis. We wanted to see if we found some sort of a structural correlate to this sensory cognitive segregation of areas. And we did indeed find that when you move along this axis and you move towards the right, the number of receptors per neuron increases linearly, and it is nicely represented in this plot here, and this increase is supported also by increases in structural changes such as larger dendritic tree in areas that are located to the right of the plot than in areas located to the left of the plot, and also more spines, okay? And the opposite situation is that when you move from the left to the right of the sensory cognitive axis, we find a decrease in myelination, which is sort of like logical, because we know that if we've got myelin, we can't really have synapses, okay? Now, we could not just, plot our areas on a two-dimensional plot as we've done here, but we could also, well, not we, Sean could, project them onto the surface representation. This is showing the Yerkes surface brain or template. And what I found fascinating when I saw this image was that these principal, or this first principal component, the one that separates the sensory cognitive axis of the brain, actually, if you look at the distribution of the blue and the red areas, it compares very well with the functional zones that Marcel Mesulam defined in, well, here I'm citing the 2000 chapter. It's one of his seminal works, but he's got so many again. So it's not just nice to see that the multidimensional scaling in the human brain and the principal component analysis in the macaque monkey brain showed us the same sort of a segregation. This is good for us if we want to do translational studies. It's also nice to see that the visual representation is also very intuitive and also helps to establish these correlations. Now, moving on to another study, this is the one that Sean was actually really interested in when he addressed me. He wanted to know the effect of dopamine D1 receptors on working memory. Dopamine 1 receptors weren't ones that we used normally for mapping procedures because they don't, you know, the changes in receptor densities don't stick out like sore thumbs when you look at this receptor. It's just a lot of red in the middle and shades of blue on the outside. So I actually didn't have D1 receptor densities when Sean approached me, so we had to extract them. But we did it, and we found that the density of D1 receptors increases systematically along the hierarchy. And then Sean used this information to inform one of his models, okay? And I'm just showing this here because, well, basically because I want to advertise the receptors a bit, and I think they're useful tools, and if anybody wants to use receptor densities to biologically inform their models, you know, feel free to ask us or ask Sean how he's implemented it, you know. But what he could find, and that's the nice thing, is he could find that the fact that his model included actual data on receptor densities or D1 receptor densities, and that we have this gradient along the hierarchy, that enabled him to explain the fact that dopamine release can shift the brain from an activity-silent regime to a persistent activity regime, okay? And it also helped him to predict that short-term synaptic changes should occur mostly in the sensory areas. And this is something that apparently had been, you know, in his field discussed. It's not something that I was dealing with. Models. I'm now moving from the macaque back to the human brain because I want to present work of a collaboration with Yasser Ituria Medina and at the time his PhD student Ahmed Khan. They used the human receptor densities for their models. They were trying to create personalized models to predict either what receptor type could maybe contribute most to the progress of the disease, and or which patient subset would maybe react better to medication depending on their receptor fingerprint, okay? And again, you know, this modeling stuff, I did read that article, you know, from front to back, and I think I tortured poor Ahmed because I kept on asking him, what does this R mean, and what's the difference between this capital R and the non-capital R and the one in cursive and not, and is this a typo or does it mean something else? Am I just not getting it? So I think he had more trouble with my questions than with the reviewers' questions, but yeah, I'm not going to pretend to really understand what he went on here. Now, I want to stay in the human brain, but I want to move to in vivo. I want to move to PET because, let's face it, if we want to talk about function, it's actually better to be able to do something with living brains because my ones are post-mortem, and even if we use my receptor data to predict why something is happening in resting state fMRI activations, it gives resting state a completely new connotation, right? Because basically what my receptors are showing is the eternal resting state. But basically it's nice, and it's also reassuring. And the PET images that you can see on the right of the slide, they're images that are very kindly being made available to the community by Rupert Latzenberger on his home page. So a big thank you to him. And what I find reassuring is that they show a we've got very low densities in the occipital lobe, and they increase on the medial surface of the hemisphere as you move rostrally. And when you look here at this fingerprint, you see we've got these areas here. They are all visual areas. They've got very low densities. Here you've got the hippocampus. The hippocampus is this structure here. So we're seeing the same thing, post-mortem and in vivo. I've got the advantage post-mortem that it's a better resolution, and in vivo it's in vivo, and you can look at time changes. But PET images, of course, bring me on to the fantastic work of that Justine made in collating this impressive collection of PET data, which encompasses now data sets from 19 different neurotransmitter receptors, and we've got mean receptor distribution maps, which represent a total of over 1,200 subjects, okay? And that's also a lot better than my N equals 4 post-mortem data. Justine, by the way, has a poster, so do go and see her. Justine analyzed relationships between structure and, or not between structure, between this PET data and function, and she could find what you see here on the left is the structural connectivity matrix of her not of her sample. Ask Justine about it tomorrow at her poster. But it's a functional connectivity matrix, okay? As opposed to this functional, it's a structural connectivity matrix in blue, as opposed to a functional connectivity matrix on the right side in tones of red. And what she found, which is also very reassuring because it's something that we've been postulating for a long time, is that areas that belong to the same functional system share more commonalities in the receptor architecture than other areas. Justine also compared PET and also PET data with the expression levels of encoding genes at the whole brain level. She found a very low correlation. There were only 2 receptor types that had a positive correlation between receptor densities and genes. She found them. Oh dear, I've got to hurry up if I want people to ask questions. This isn't a question of maybe, you've got different subjects. One of my postdocs, Ling, was able to prove this in tissue samples from the same brain, that we do have a relatively poor correlation between receptor densities and the expression levels of their encoding genes. And I'm now going to move back from the human to the macaque monkey brain and 3D reconstruction of autoradiographs. And this is work carried out by Thomas, who did his PhD at McGill with Alan Evans, and he then moved on to do his postdoc, or his first postdoc with me, and he's now at the Child Mind Institute. And it's Thomas's poster tomorrow, so go and see it. I hope this is enough advertising for you, Thomas. Now let's move on to what you did work-wise. Thomas had a lot of problems reconstructing the 3D autoradiographs, and basically it took me 20 years to find somebody who would take on this task. And you might wonder, well, why is it so difficult? Why was one of his problems nonlinear deformation of brains? I mean, he was working at Alan's lab, and Claude Lepage works there, and he reconstructed the big brain, and the problem's been solved again, right? But the problem is that for receptor autoradiography, we need fresh brains, not fixed brains. And for those of you who haven't worked with brains, a fixed brain has the consistency of an eraser, but a fresh brain has a consistency which is somewhere halfway between jelly and an egg, okay? So this here, meat, MR2 didn't always look like this. MR2 did look like this at some stage or other, right? So poor Thomas had to bring MR2 from the situation that you see at the top of the slide back into the situation that you see in the bottom of the slide. And to make life more difficult for him, for technical reasons, to visualize the receptor autoradiographs, we had to cut each slab into, or each hemisphere into slabs. So you can see here, this isn't an entire, or it is an entire slab, but it's been cut, it's been sliced, right? So he had tissue slabs, there were different deformations. It was a big mess, really. So different kinds of deformations in different parts of the brain. And then he had the added problem of really large variability in pixel intensities, because we've got different receptors and they're all characterized, by a different distribution in the brain and different absolute densities. And on top of that, as if that weren't enough, well, I also wanted him to please reconstruct the cell body and the myelin stained sections, right? So to do this work, Thomas created a wonderful pipeline which he called Brain Builder. It's just up here as a placeholder. Go and see Thomas at his poster, and he can tell you all about it. And he reconstructed the human brains. And this here is just an overview of the human brains, right? He originally created this pipeline for the human brains. And this is a wonderful animated GIF through the macaque monkey brain, which is also showing these receptor densities. And now we've quantified a bit, so which receptors across the entire brain help us, maybe identify areas. And we find that at the right of the slide, the areas that are most different, we've got the serotonin receptor, which has a super distinct laminar distribution pattern. It has the highest density ratio. And with highest density ratio, I mean that it's the difference between the area with the highest and the lowest density of that area. So it's by far, the highest one. Kainate receptor densities, they're my favorite ones, because they're the ones that show a high granularity. They're the ones that show the most borders. And M2 receptors, you've seen them earlier on. They are super interesting because they highlight the functional, the sensory systems, and they are also important, or they're also interesting because they show a variable laminar distribution pattern throughout the cortex. And this plot here, by the way, is really hot off the press. So Thomas created it the day before yesterday. Thank you. And with this, I'd like to finish just my take-home message. There've been several talks that have said that we have to integrate, the functional data, work out which one of all the different parcellation schemes that exist is the true one. And I think that that's a really good idea. The slight caveat, I think we shouldn't just use the functional parcellations, we should also include the structural parcellations, because without structure, there is no function. Okay? And with this, I would like, I'm come to the most difficult part of my talk today, and I hope I say it correctly. Kyung chung hai tsu su seu kam saham nida. Thank you all for your attention.
Thank you for that absolutely wonderful talk.
Thank you.
So we have time for a couple questions. The first one is, how stable are these receptor densities throughout the lifespan and also across different healthy and clinical populations?
They change throughout lifespan. There are studies which have dealt with that. The problem is there are no whole brain studies, so we know for certain brain regions that certain receptor types are higher in infants, and then they change up to adolescence, and then they remain constant. Other ones start to decline later on in life, so that there is no systematic answer. It depends on the receptor that you're interested in. And differences between healthy and controlled subject, healthy and disease, millions of differences. They affect different receptor types, different neurotransmitter systems, different brain regions, depending on the disease. And it's normally so that you rarely have changes in a single receptor type or a single neurotransmitter system. You normally have changes that affect different neurotransmitter systems.
Thank you. And we're going to do one other question. So you talked a lot about the different resources and tools and atlases that are available. So do these resources, tools, atlases include also data on the receptor densities within the cerebellum, subcortical structures, brainstem, and so on?
Not yet. I do have data for the human cerebellum and brainstem. I'll get there, or I'll look for, if anybody has a postdoc or a PhD student that wants to do, go ahead. I have been concentrating on the cortex. I am now moving down into subcortical structures. There's the thalamus, amygdala, I'm doing the claustrum, and then we'll move down a bit more. So yes, they will eventually.
Of course. Thank you very much. Let's give her another round of applause. And just a reminder to everyone that club night is tonight from 8 p.m. to 11 p.m. So we hope to see you all there. Thank you.